So this is, let me just key up what we're going to do for this next half hour. We're going to take time to review the results of the testing panel. We're going to be showing you in aggregate the results of the first 175 that attended this conference and the results of those genotypes. Dr. Brown and I will be splitting the presentation. I'm going to go over some general things first regarding the report, and then he's going to jump into the more interesting things, which is the aggregate genotype results, to understand what we look like as a group here genetically. And I want to give a special shout out to our students who worked so hard at putting this together. Two of the graduate students really worked hard at getting the DNA collected and getting kits out to you guys. And one of the graduate students really worked hard at putting together the slides and crunching all the data. So I really do appreciate what they have done. I also want to make sure I thank OneOme, who contributed the genetic testing for the conference. So hopefully y'all made it over there to discuss things with them. So this is our outline. So I want to just start for the people who didn't go through the process of getting pharmacogenetically tested. DNA was taken from individuals that wanted to be tested from a buccal swab. So a kit was obtained by the individual. They did a buccal swab. That swab then was sent back to OneOme for the genotype testing. OneOme has 82 different genetic variants that they call on this particular panel, and across 27 different pharmacogenomic genes. So the way it works is that the calls from the TACMAN assays will produce a genotype. The genotype then is converted into a phenotype, and OneOme has a proprietary system by which they make those phenotype calls. That's the way most of all the commercial companies operate. They have a system by which they convert genotype to phenotype. So this is an outline of the report. So this is what the report sort of looks like. I'm holding it up here, so you who picked it up, it should be this white piece of paper. It's called the RightMed Comprehensive Test Report. And you'll see right away that there is, in general, there's 3 different categories that these drug-gene interactions fall within. One is a major gene-drug interaction. It's highlighted in red, means be cautious. A moderate gene-drug interaction, which is the yellow, the little yellow triangle, and then minimal drug-gene interaction, and that's represented in green. So most of the companies, the commercial companies that do pharmacogenetic testing, they do use this sort of stoplight approach, the green, yellow, and red, and it's to simplify or at least alert the clinician as to what are the most concerning results or what the results are that aren't concerning. So the stoplight approach is common. You'll also see in your report on the first page that there's a legend, and it tells you what the interaction might be, what the gene-drug interaction might be. This little, for example, the first icon is this little blue box with a positive, which means increased exposure. So that would be most likely somebody who's a poor metabolizer. They're going to have accumulation of the drug, and they're going to be at risk for greater adverse effects. This is an example, just a cut and paste from one of the OneOme reports. So you can see for this particular sample patient or individual, there's a number of major gene-drug interactions that were identified. Morphine was one of those. There's a number of moderate gene-drug interactions that were identified. They're listed there. And then there's also some that were minimal drug-gene interactions. The list is much longer than this. I just a cut from a few of the pages. They also go on in this report to explain the gene and phenotype summary. I believe that is maybe about the page maybe page 5 in your report, that it is listed gene and phenotype summary. So these are your results, and it gives the genotype, which is in this case, let's just do CYP2B6. this particular example, this individual has the genotype star 1, star 5, and the phenotype for that particular genotype is an intermediate to normal metabolizer, right? So you'll see that there's that gene and phenotype summary for every gene, and every variant that was tested, and it gives you some idea as to what the implications are for that particular gene. So I just want to pause, and I know for some of you this is going to be too simple, so you can, just zone out for a minute. But I do want to explain something, because this sometimes is kind of confusing about interpreting genomic results. So most of the genetic variants that are pharmacogenomic variants are SNPs. So they're single nucleotide polymorphisms. Not all of them, but most of them are single nucleotide polymorphisms. And what that means at the genome level is that this would be the DNA strand, and you have a whole bunch of A's, C's, T's, and G's that get strung together. These nucleotides get strung together in certain sequences that make us all unique. And the top sequence that I have represented here on the slide is the normal or the wild type sequence. But in the case of a single nucleotide polymorphism, this would be that T, the T that's in red. So normally at that position you would have a G, and in this case, this sequence, it was for some reason a T. So that's a G to T conversion, so the variant is a T. Each one of these variants, and there are many across the human genome, is given a unique number. These are called RS numbers. They all have a number, and that's to assure consistency across laboratories, across the way we interpret these. So every one of these single nucleotide polymorphisms is given an RS number. Now, one thing they've done that's unique, this is frustrating to some folks, but it's unique in pharmacogenomics, is they use this star allele nomenclature system. It's really to help simplify interpretation and our discussion about it. So they have all given these RS numbers all star designations. So in the case of this RS 776746, this is a SNP. It's also called the star 3 variant. So these are the same, represent the same genetic variant. It's just the star 3. It's a lot easier to say star 3 than 776746. And then another, just a small point, is a star 1 designation. That's wild type, meaning that's the most common of the alleles that occurs at that particular position. Here's an example. Here's CYP2C9. Here, and this is the star 2 variant. So this is RS. Here's the RS number. Here's the gene name. Here's the nucleotide change. So at the position 430, there it is normally a T. It's been converted to, or it's normally a C. It's converted to a T. Here's the star nomenclature. They've labeled this one star T, or star 2. This is the enzyme activity as a result of this particular star allele is inactive enzyme. So you can just guess from that you're probably going to be individuals that carry one or more of these are going to be poor metabolizers. So here would be an example genotype. So a person might be CYP2C9 star 1 / star 2. Star 1 means that they carry the one strand of their DNA carries a normal or the wild type allele. The other strand of their DNA is star 2, which carries the mutation. You can also carry 2 mutations, so that in an individual that would carry that would have a star 2 genotype, meaning they had two genetic variants. And then the last thing I want to talk about in this regard is the terminology homozygous and heterozygous. Of course, those terms are used all across genetics, but in particular, I just want to go over 3 examples. An individual who has a CYP3A5 star 1 genotype would be homozygous for 2 wild type alleles. Homozygous meaning that both of the alleles are the same. So homozygous for 2 wild type alleles. An individual who's star 1, star 3 would be heterozygous because they have 1 wild type and they also have one variant. And a star 3, they are also homozygous, but they're homozygous for the mutation. Okay, so just a little bit on that. Okay, now jumping in, this is where when I look at reports, I like to jump to this page immediately. But this is the page that actually gives the RS number and the alleles that were tested, and then the result. So in this particular report, this is a CYP2C19 test, and within CYP2C19, you can see that on the OneOme panel, there is 5 different genetic variants that they test for. The top one is the star 17 allele, and at position -806, it's a C to T conversion. And then if you go to the right and you read the genotype that was measured, this individual is a TT. So this person has 2 variant alleles at that position. That's why they were called the star 17, star 17. So they're CYP2C19, 17, 17. Okay, you do the same thing for CYP2C9 or star 3. This individual has a star 1, star 3 genotype, and you know that because of this particular RS number 1057910. Okay, and on and on. Now, for the HLAs, those are reported as positive or negative. Those are different kind of assays that are done to develop that, but those are just reported as positive or negative. So now, in regards to all of you who underwent the genotyping, these are the different. This is the on the left-hand side is the distribution of the genotypes that were screened. So you can see from this group of only 175 individuals, there were a lot of different genotypes that were identified. If you look at then at this distribution, those genotypes then were converted to phenotypes, and there's even more phenotypes. So there's a lot of different genotypes in this room and a lot of different phenotypes in this room. And Dr. Brown's going to talk a little bit more about those in detail. This slide, I know it's a little bit complex, but bear with me on this, that if you just look at CYP2C19, of the 175 individuals that are represented here, there were 9 different genotype combinations, which resulted in 6 different phenotypes. So there are 6 different ways in which this group metabolizes substrates by CYP2C19. If you look at CYP2C9, there were 8 different genotype combinations, 4 different phenotypes that were produced from there. CYP2D6, you know that that's a massively complicated gene, a lot of different alleles and haplotypes. There were 49 different genotypes that were identified in our population here, resulting in 6 different phenotypes. And then for TPMT, and of course you know that's one of the genes that regulates the metabolism of 6-mercaptopurine. There were 3 different genotypes that were identified in us, and resulting in 3 different metabolism phenotypes. So I always love looking at this kind of data because it just goes to show that we all have something we all have some sort of variation within us. So this is just looking at 12 genes, and these are the 12 genes that come from the CPIC guidelines. So these are genes that are considered to have actionable mutations within them. Of the 175, there were only maybe about 5 of you that only had 2 mutations. There was about 20-some individuals who had 3 mutations, about 40 individuals that had 4 mutations, over 60 of you had 5 mutations, there's about 35 of you who had 6 mutations, and about 15 of you had 7 mutations. So you can see it's not just sick people who have mutations. Even a healthy group like this is going to have pharmacogenomic mutations. And hopefully that could be something, I've always promoted that we need to do preemptive testing where these kind of genotypes would go into the electronic medical record. And even though right now I don't need these genotypes, someday I will. And then they would be placed there for me. Oh, right, I'm gonna stop right now here and let Dr. Brown talk about the rest of it.
All right, thanks, Dr. Jacobson. So for those of you I have not met, my name is Jacob Brown. I am an assistant professor up on our Duluth campus with the College of Pharmacy, and actually I get to moderate the next section. So I'm going to finish up this part and then roll right into the afternoon session. But so we heard a lot about ethnicity with Dr. Karn's presentation and also Dr. Johnson. So within our 175 individuals, so again, the first 175 people that registered, and this is self-identified ethnicity. So Dr. Karn's very nicely discussed some of the limitations of that, but of those 175, around 75% self-identified as white or Caucasian, around 50% as Asian, and then the remaining as either unknown or not provided, Hispanic or Latino or Jewish descent. So this is going to be important because we broke out some of our results comparing both individuals with Caucasian descent and then Asian and showing some of the differences between the allele frequencies in some of these genes. So this figure is probably a little bit hard to read up there, but it's from a publication last year in Clinical Pharmacology and Therapeutics. So this is actually an excerpt of this, so the figure itself is even bigger if you want to go take a look at it. But it really nicely shows what the allele frequency is of some of the different variants that are identified in general population. So on the top here, we've got ethnicities in the columns, and then we've got the different genes, so limited to the 4 cytochrome P450s there on the rows. So I think what stands out the most in this figure is that on the top part, so we've got CYP3A5. Dr. Jacobson already talked about that a little bit, but we see here that in essentially non-African populations identified up there, nearly 100% or very close have that star allele or that star 3 allele present, and we're going to see this very clearly within our Caucasian population. And then we have a couple other alleles that are more frequent within African populations. As we go to CYP2C9, we see a little bit more just variability between the different allele frequencies observed within that gene, but then across different ethnic populations. CYP2C19, which we're going to talk about as well a little bit more in depth, has probably even more variability within there. And then as Dr. Jacobson talked about, I think there were 49 genotypes identified for CYP2D6 just within our 175. So CYP2D6, you can see, has a whole host of different allele frequencies, and it varies significantly depending on what ethnicity or population we're talking about. All right, so I kind of get the fun part of this presentation because I get to share what our specific gene and allele frequencies look like. So we're going to start out with HLA-B genotypes that is included on your OneOme report. So again, you can kind of follow along with this if you'd like, or certainly go back later. So on top there, if you're listed as negative, it means that you do not have that variant. If you are positive for that, that implies that you are at an increased risk for some of those like Stevens-Johnson or toxic epidermal necrolysis that were previously nicely covered. So we see that on the blue line there on top, over 90% of individuals of white or Caucasian ethnicity do not have any of these HLA alleles that were tested for, so they would not be at increased risk for some of these medications. When we look at the 5801 HLA, we see that it's not common or not seen maybe in one individual within our whiter Caucasian population, but nearly 1/3 of our individuals of Asian descent. So allopurinol is one of those medications commonly used that potentially individuals with that variant could be at risk for some of those skin reactions. When we look at the 5701, and again, Dr. Karn's alluded to the host of different HLA variants that are out there. The 5701, we see relatively similar frequencies there, and then for the 1502, we only see that again in Asian populations. For our 100 and it's probably 160 or so individuals that I self-identified as white or Asian within this population. So with the takeaway point that individuals of Asian ancestry are more likely to carry that variant and thus be at an increased risk for some of those very serious skin reactions if they were prescribed or taking some of those medications. All right, so then we have CYP2C19 genotypes by race. CYP2C19, again, is relevant for proton pump inhibitors, some of the antidepressants out there. Some of our pharmacy students should probably be able to talk about some of those. But when we break this down by genotype, we've got star 1 up at the top. So we have individuals who are homozygous wild type. That accounts for about 40% of our white or Caucasian population, and only about 20% of individuals that are Asian. So meaning that essentially everybody else has either slightly increased, slightly decreased, or significantly decreased CYP2C19 activity, which, as you can imagine, results in quite a bit of variability in terms of plasma concentrations that we observe with some of these medications. So notably, if we look at the star 1, star 17, we see that in individuals of white descent, nearly a quarter of individuals have that star 17 allele, which denotes increased metabolism. So those individuals are categorized as rapid metabolizers. And less than 4% of individuals of Asian descent we see that in. So with the star 1, star 2 here, and then if we sort of include the star 3 allele in there, the star 2 and star 3 alleles are more commonly seen in Asian populations than Caucasians, which is very nicely displayed here. So we see that 42% of Asian individuals carry that star 1, star 2 genotype, as opposed to only around 20% of individuals of white descent. And we see that for the individuals that have 2 variant alleles, or would be homozygous variant there, so poor metabolizers, we see very limited in white or Caucasians, and then greater than 20% of individuals of Asian descent. And then again, if we look at that star 17, we did not identify that in anybody of Asian descent there, but somewhat frequently in a white or Caucasian population. So as Dr. Jacobson and several others have mentioned, when we talk about genotypes, we talk about how that is translated into a phenotype. So on your OneOme report, there are 6 different categories. I would just point out that if you're looking at a CPIC guideline, there are 5 categories. So CPIC includes all of them listed up here, but they do not include the intermediate to normal category. If you find that discrepancy. Since some of those genotypes fall in the same phenotype category, we see that in an Asian population, greater than 50% of individuals are considered intermediate metabolizers. And as Dr. Johnson very nicely talked about this morning, with clopidogrel, that constitutes a fairly significant portion of individuals where if you're following CPIC guidelines, your recommendation is to use something other than clopidogrel in that population. We look at normal, only about 40% of white or Caucasian individuals were considered normal for CYP2C19, and then about 20% for Asian. About a quarter individuals who are white are considered rapid or increased metabolizers there, and then again, we only identified poor metabolizers predominantly within the Asian population, but does make up about 25%. So pretty significant variability, which has pretty direct implications within a lot of the CYP2C19 medications that we use. All right, and then so for the CYP3A5 genotype, this one is interesting because the vast majority of white or Caucasian individuals carry the star 3 allele, as was shown on that previous ethnicity side. So fortunately, our 175 participants kind of follow those allele frequencies, so it validates kind of what we expected to see. But we see that greater than 96% of white or Caucasian individuals are homozygous for the star 3 variant and are considered poor metabolizers of CYP3A5, and that is markedly different on this slide as compared to Asian individuals, where about a third are intermediate, and then about a little close to 10% are considered normal metabolizers. So within the star 3A5 genotype, when we talk about the star 3 allele, it's one of the more commonly seen ones, particularly within a white or Caucasian population. All right, so we're gonna fairly quickly go through 2 CPIC guidelines and just talk about how the phenotype groups that we saw within our 175 participant translate to some of those recommendations in dosing changes from CPIC guidelines. And I'm not gonna talk too much on this because obviously our next speaker is the director of CPIC, so she can speak much more to it than I can. So for thiopurine methyltransferase, or TPMT, this enzyme is involved in the metabolism of 6-mercaptopurine, azathioprine, 6-thiopurine. So on the top here, all CPIC guidelines follow the same format, where on table 1, they show you what the likely phenotype is, what examples of some of those genotypes are, and then what those diplotypes look like. And then table 2 gives you what the recommended dosing is by phenotype. So for this example, we've got homozygous wild type. So essentially, if you're star 1 for TPMT, which most people within our 175 are, the recommendation for all 3 drugs is to give the full normal dose that you would start with anyways, even without this information. If you're a heterozygous, so if you have 1 normal, 1 loss of function allele, the recommendation would be to reduce that for 6MP and then azathioprine by 30 to 70%. And then if you're a homozygous variant, the recommendation is to drastically reduce the dose and the frequency, or consider an alternative agent, because those individuals would be at a pretty increased risk of some toxicity from those medications. So when we look at our allele frequencies for TPMT genotypes within our group, we see that so for the star 1 was the vast majority of individuals. So most people within that 175 would be recommended to take the normal starting dose and go from there. There's about, I think, 5 to 10 individuals that fall into this increased risk category where they have, where they're star 1 and star 3. So those individuals in particular would then be recommended, if starting on one of these medications, to have their dose reduced by to 30 to 70% of the starting dose. All right, and then we're going to finish up with with an example of CYP2C19 with both clopidogrel and then voriconazole. So I would just note that voriconazole is one of the few CPIC guidelines that has unique pediatric recommendations. So it varies with rapid metabolizers, but it is certainly helpful for pediatric patients. So for clopidogrel, Dr. Johnson already spoke of this, but essentially if you have 1 or 2 of the reduced function CYP2C19 alleles, your recommendation is to have an alternative to clopidogrel. And then for voriconazole, essentially if you're at the ultra-rapid end of the spectrum or you're at the poor end of the spectrum, the recommendation would be to use an alternative antifungal agent there. So when we look at our population, if we think about this in the context of clopidogrel, and we look at the phenotypes here, we have about 40%, again, that were considered normal, and we saw this before when we broke it out between white and Asian populations. Close to 25%, 25 to 30% are considered intermediate metabolizers. So about 35 to 40% of the population within our 175 people that were genotyped for this conference would actually be recommended for an alternative agent of clopidogrel if they were in that clinical situation. And then in the context of voriconazole, again, we're looking at sort of the 2 ends of the spectrum. So we'd have individuals that were rapid metabolizers, so that made up about 20% of our group there. And then ultra-rapid or poor would make up another about 10%. So about a third of individuals would be recommended from CPIC guidelines to include or to use an alternative antifungal agent. All right, so those are all the results that we have. If anybody has any questions on those, you can certainly contact us. But I think we'd be more than likely to share those slides. Zach actually put a lot of those together, so you can email him or myself. So quickly in summary, I think we really are trying to make the point here that most individuals carry variant alleles relevant to medications. I think the median there was between 4 and 5 within the OneOme panel. Variants can really range anywhere from poor metabolism to ultra-rapid metabolism, and this is going to have pretty different implications depending on what type of drugs we're talking about. 1 gene can be relevant to more than 1 medication. So our example was CYP2C19. There are several CPIC guidelines with CYP2C19. For different medications, which gets to Dr. Johnson's point that when they had that information, many individuals were then later on put on additional medications where that information would be very useful to that individual. The cumulative number of variant alleles matters, again, towards metabolism. So if you have more than 1 variant allele, this can kind of push you in one direction or the other. Multiple variants and haplotypes in a single gene can be relevant. So we looked at the example of CYP2D6. And then variants in multiple genes can also be important. So we're certainly going away from that 1 drug, 1 gene format and trying to include as many genes as within reason that can explain the variability that we see within a general population. And currently, I think CPIC guidelines are available for 37. Kelly will certainly correct me if I'm wrong on that on the next presentation. So if you have questions about your genotype report, again, you can talk to individuals back at our personalized medicine table. Sam is manning that at the moment. And then certainly questions about the report. Obviously, you guys got those from OneOme. We, again, would just like to acknowledge Zach Rivers put in a lot of work to kind of put these statistics together for us, and then helped out quite a bit with the slides. Sam and Jay, graduate students, also helped out a lot with the sample collection process. Diane Blumenfeld of our staff that helped out with this, and then Ross Higgins from OneOme, who helped with the sample collections and then running that and then returning results.